Good afternoon to everyone present in the audience. We'll be beginning shortly. Thank you. Good afternoon and welcome to the second session on the webinar on battling COVID-19, its challenges and opportunities in the healthcare and biopharma sector. The following session is going to focus on the healthcare and biopharma sector and feature established experts of the field sharing their insights. Our first speaker for the session is Mr. Mark Stevens. Mark Stevens is a partner in the life sciences practice at Guidehouse and is responsible for leading their efforts to expand and build consulting capabilities in the United States and in India, respectively. With more than 20 years of experience in the pharmaceutical and biotech industries and extensive experience in Salesforce effectiveness, data analytics and visualization, corporate strategy, and other aspects of commercial excellence, Mr. Stevens has played key leadership roles in several organizations, including Novartis, Bristol Myers Squibb, Johnson & Johnson, Sanofi, and Publicis Health Solutions. He is also co-founder and president of a non-profit organization called the South Asian Pharmaceutical Council and is working towards improving healthcare in South Asia. Mr. Stevens has extensive experience in product launch research and launch metrics generation and tracking for a wide range of therapeutic areas. He is also an author of numerous blogs on pharma, such as New Commercial Models, Participative Medicine and Patient Engagement, Precision Recruiting, Organized Customers, and Pharmacogenomics. I would like to hand over the mic to Mr. Stevens.
Thank you so much. Can you guys hear me?
Yes, we can.
Okay. So thank you so much for this introduction and happy to be here. Can you see my screen as well?
Yes, sir.
Okay. So I'm glad to be part of this webinar. I'm glad to be actually honored to be part of this very distinguished panel you guys have put together. And I'm going to be quick. I have about, I think, 35 minutes to go. So what we're going to talk today is three things, a little bit about the virus, a lot about the biopharma response to the pandemic, with special reference to vaccines in development, antivirals in development, looking at what's happening with clinical trials across the world. I think I'm going to leave you guys with the challenge of what can all of us, including people who are part of this panel, do about the pandemic. I think there is many of us who are involved in research, some of them involved in biopharma commercialization. What is everyone's role? This is a pandemic. I'm also going to tell you a little bit of a disclaimer of what I'm not going to cover. I'm not a physician or a lab scientist, so don't expect a very in-depth recommendations of molecular structure or, binding properties and things like that. So number one, it's not relevant for this talk. Number two, I'm not an expert. There are two things I'm not going to cover, anything to do with med tech and with other than Western medicine. I think there is panel members who are way more qualified who are going to talk to you about Ayurveda and med tech, which is tomorrow. The other thing is I'm not going to talk about I'm not going to give you any recommendation or talking about economic impact. I think that's a topic by itself. I'm going to turn off my video so there's more emphasis on this one. I'm a partner in a global consulting firm. We have various verticals, life science being one of them. So we look at the life science part of this pandemic differently. And in full disclosure, we're actually consulting with one of the companies which are coming up with a vaccine for COVID-19. And I'll talk a little bit about that. I cannot divulge a company, but we're very involved in the commercialization of that vaccine. If I can switch. So what is COVID-19? I think with a very quick, I think the whole coronavirus family of viruses is very common to us. Many of us would know, some of us probably know, that's the same family of viruses which causes us a common cold, which most of us, all of us have experienced throughout our lifetime. It is the same family which came up with the SARS, the severe acute respiratory syndrome, or the MERS, the Middle Eastern Respiratory Syndrome. In 2019, towards the end of 2019, a new coronavirus was identified as a cause of disease, and it's believed it originated in China. Although it's believed to be originated from bats, the exact source of SARS-CoV-2, animal reservoir, and enzootic patterns of transmission still remain uncertain. There's various theories, but none of them have been confirmed. Technically, the disease is known as SARS-CoV-2, and in March 2020, the World Health Organization declared COVID-19, the new name, outbreak as a pandemic. It's very interesting. I'll give you a brief idea of what this pandemic has done, and then we'll go quickly into the biopharma response. This deck is chock full of information. We might have at the end, depending on time, I want to be very cognizant of the speakers who are going to follow, making sure that they have the right time, and I stick to mine. There might be time for a question or two, and we'll talk about that soon. December 2019 was a regular year for many Indians and many across the world. And if you can see, in January and February and then in March, this became very quickly a disease which to be reckoned for. And the first death in the US was in February of 2020. And in February of 2020, I think WHO named the disease virus as COVID-19. And it's fast-tracked to October 1, which is 3 days before. We have about 1 lakh deaths in India. So this talk is mostly global. Wherever I have data on India, I'll include it to make it a little more pertinent to the discussion and to the audience. And I think this is going to be interesting because it's if you look at pandemics across the life spectrum of human beings ever since we were here on the earth, there's various pandemics. I think not many people have lived through a pandemic, but quite a number of them have lived through a pandemic. And actually, the good news for us is, all of us who are listening to this are living through this pandemic. I think it's something you can talk to your children and grandchildren at some point when next year this will be a disease like the common cold. Unfortunately, there's been 33 million cases across the world with COVID-19. And you look at this map, it's actually really sad that at one point, every part of the world is affected. Very rarely this happens that, sometimes a flood happens, sometimes, Ebola was in Africa, but not a disease which really everyone in the world was really looking for. In India, there's close to 6.5, I think total deaths increased, and this is the data from September 29th, last month. And I think you guys have crossed 1 lakh. In the US, there's been 7.2, and total deaths have been about 205,000. Brazil being the third. We'll watch these 3 countries as we go, but I think the reason why it's a pandemic is because what you see in this map. Fortunately, there are some places which have not been as devastating as many other states and countries. And I think this is an epidemiology which is going to be interesting. Nothing has been set in stone in terms of age, in terms of geographic distribution, in terms of deaths, in terms of infection, in terms of herd immunity. A lot to be studied about this disease, because right now it is what it is. And that's one of the challenges I'm going to force you guys later on in the presentation. So I'm going to keep a time. Okay. So let's talk very briefly about the virus. I think most of this group knows that the virus is a non-living by itself. It typically contains a protein coat surrounding RNA or DNA core without a semi-permeable membrane. They are incapable of growth unless they multiply on another host living cell. So it's interesting that deadly as a disease done by a virus can only live with someone else. You know, the reason why it's called corona is because that's how it looks. It looks like a crown under the microscope. In biological terms, coronaviruses are called impositors because they enter the body as very safe, but I think once they attach and they multiply, there's no stopping to them. So look at it, it's an invisible virus, only can be seen under the electron microscope. And it's non-living. Imagine the devastation of this one small creature can do as a non-living and can only living on a host and invisible. It's amazing that the entire world of 33 million people have come in contact with this stuff, and wars can happen and natural calamities can happen, but they're all physical, and this one doesn't seem to be physical. And that might be one of the reasons why the disease is growing so fast. We'll talk a little bit about this, but I'm not going to talk about the morphology and physiology of this stuff. Putting in perspective with death toll, and I think this is an older slide. I'll be showing you slides from various dates, but they all, dive into the story. So right now we have about 1 million people who have died from the coronavirus worldwide. Put that in perspective with the Black bubonic plague of 1347 and 1351, 200 million. Smallpox in 1520, 56 million died. Spanish flu 1918, 40 to 50 million died. If you keep looking at this, we are somewhere around this Asian flu and the Russian flu, about 1 million. We might be going across and be the third in the bottom line after this century great plagues in the 1665. So in perspective, as it's not the smallest virus which is causing the smallest damage, it's actually increasing, but not as bad as what the world has seen before or what our forefathers have seen before, where the entire world has seen 200 million deaths. I think we'll catch it way before that becomes larger. So let's look at what, to your left is what I gathered from the New England Journal of Medicine. To your right is the Centers for Disease Control in the US. What are the guidelines in terms of the virus? So the virus is thought to spread mainly from person to person. Between people who are in close contact one another, it's about 6 feet. And you'll see the reason why the 6 feet is a technical distance. The respiratory droplets produced when an infected person coughs, sneezes, or talks. And these droplets can land in the mouths of noses of people who are nearby, who can be inhaled into the lungs. And COVID-19 may spread by people who are not showing symptoms. So on your left, you're seeing by far, what's a common understanding of how long does this virus survive on certain substrates, paper and tissue to outside of surgical mask. So in terms of cleanliness, in terms of washing your hands, in terms of just washing and making sure the touching and all this stuff is there. There are some guidelines. I think you need to be careful. What's interesting is just because you wear a glove doesn't mean you're immune from that. You can touch, you can touch any one of the one on your left and the virus is there. I think the best thing is the mask part, and washing and just be cleaning and be careful about the hygiene is important. So here's how the Ebola worked out. In Ebola, I think it's 3 feet. I mean, I'm glad that is not an issue. 6 feet is actually a safe distance, but not good enough to be inside an elevator. But I think this is by far the best preventive measure, to be 6 feet or more from any person. And that's why the mask is important. I think this is by far the best preventive measure, to be away from anyone, not knowing whether they are COVID positive or not, and making sure that even with 6 feet, there is a chance that you might get it, and protecting yourself with a mask. So how can what can one person do? And it doesn't look as bad, but every person can infect 2 to 3 people on COVID-19. In, in perspective, compared to measles, mumps, rubella, polio, and smallpox, it's not that infective, but it's still, because there is this distance feature, and I think we haven't hit herd immunity where 50% or more of the population are immune to the disease. So herd immunity is defined, many people are different, either through vaccination or through being non-symptomatic. The more immunity people get in a population, the more less of the infection. There are more less and less people get infected. And that's the whole idea of this vaccination as well, before therapeutic interventions in terms of drugs come into place. So let's look at what happened in the US. In the US, we saw this big spike in May of 2020, and then it was managed well, it came down. And then we saw the second spike of where the economies were open, with special reference to states like North Carolina, Florida, and California. And then we see this dormant period. We're expecting to see this dormant period for a couple of reasons. There's going to be some experimental drugs. But more so, I think the first vaccines, at least the mRNA vaccines, are going to be launched in the US late October and early November. Pfizer, if you looked at the news yesterday, is going to open up the trials of their phase 3 COVID-19 vaccines in 3 weeks from yesterday. So by end of October, you're going to have an approval, or maybe not an approval. They're expecting approval, which is going to provide some sort of immunity. And then comes the winter period in the US, and they expect a spike. Winter period is a spike for all viruses, influenza, common cold, everything, because it gets cold, and we expect that to be high. But I think over that, early next year, as the vaccination grow, we expect this to be plateaued, provided everyone gets vaccinated. And that's a question we need to ask ourselves, and then I'll answer you what is the current opinion on that. The unfortunate part is, look at India. India is green. India is spiking at a never-ending rate of 1 lakh new cases per day. And I think, and this is just known statistics. We'll come through later. I'll talk to you later about the latest ICMR studies which have been done, which talks about what statistics you see is far below what's really actual, based on some survey results they have seen, but we'll talk about it. But I think if you really look at the fatality ratio, which is the important thing, is coming down. I think physicians are much better at managing the disease now. And but the unfortunate part with India, the daily positivity rate is going up, and is going up and up. I think that's a cause of concern. We'll talk about when that will happen. The interesting part, between India and the United States account for more than close to 40% of the cases in the world, followed by Brazil, Russia, and Colombia. This is a staggering statistic, within the United States, and I think my assumption is India will overtake US in the coming weeks, and very soon India and US will account for 50% of the cases. So what happens to what is happening right now? I think as US, as much as it looks as one of the highest in terms of number of cases, the rate at which the cases are going, as of October 1, India stands to be number 1, both in terms of the number of absolute cases going up, and in the 7-day average of the number of COVID cases going up. I think we would like, all of us on this call would like to see that this statistics looking much better. And a lot depends on, what Indian and the Indian government and Indians as citizens do to manage this virus. India is not among the top 10 in terms of deaths per 100,000 population. That could be a couple of reasons. One, because the population is so high. Number two, the testing rates and the death rates by itself may not be that bad. United States, even though it's number one in the number of cases in terms of death, it's number 8. You'll find countries like Belgium, United Kingdom, Peru, and Spain and Italy being much, much higher up in terms of death rates per 100,000. These are actual statistics based, as of 27th. Look at the spike in India in terms of the total number of cases by day. And it's actually linear. It hasn't shown any slowing down. If you look at the same 6-month mark here for the US, you can see that there was a slight flattening of the curve, flattening of the curve from here and then from here. You don't see that in India. I think that's some cause for concern. Same thing with deaths. I think you're spiking in deaths, whereas US is seeing some trough in death. And you really look at, I've managed to have these two slides at 100K. I think in terms of India, you've already reached far higher than I would imagine any country, even the US has not reached in terms of the rate of new cases. Something to think about, and I think if you really look at cumulative number of cases by number of days since 10,000 cases, this is the patient count on your left and the x-axis is the number of days. India is growing exponentially, Yes, US is on top, but US is flattening. India is growing, and so are the other countries like Brazil and others. But I think in terms of the spike, India seems to be at a much dangerous level in terms of the number of cases. So what's more interesting is also the age. If you look at India, there's 75% are below 74 years of age. Actually 75, there's only 10% above 75. If you really look at the US, if you look at 75, there's over 60%, close to 60% above 75. So this is a very interesting statistic. There are more younger people who are dying in India than seen in the US, which has been the case. I think we'll talk a little more about the challenge about understanding this phenomena. Is this true? What's also not here, but I'll talk to it. In the US, there are more males who are affected by the disease and who die from the disease. In India, it's the reverse. There are more women and ladies who are doing that stuff. Something to think about, is that an anomaly in the US or India? Is India just because the median age is 28, in the US it's 38? Is that more older people in the US? But it doesn't really talk about why more and more young people in India are affected and die by this disease. Something I'll challenge you guys to think about in terms of epidemiology, why this is happening. It's no one knows. There's a lot of unknowns in this. There's a lot of anecdotal information, but there's no peer-reviewed journal which provides you some answer for this types of questions. So enough of the disease. We went through what the virus is about. We talked about how the world is affected. We talked about special reference to India and the US. We talked about the differences, how India is different from the US and the rest of the world. Now we'll talk about prevention and therapeutic intervention. So quick facts, and I think the highest infected countries: US, India, Brazil, Russia, Colombia, Peru, Spain, Mexico, and Argentina, and South Africa. Globally, there are more than 33 million cases and growing much faster in India. There has been no approvals of vaccines or any therapeutic drugs for this as now. There's been 5 drugs which have been authorized, not approved, authorized for emergency use, but there are quite a number of them going through trials. Most of them are existing antivirals. A typical vaccine, if you look at Merck's timeline, we'll come to that soon, takes several years. But I think an opportunity exists. There are several different types of vaccines you can do. And I think to kill the pandemic, there are much promising vaccines which can be developed very, very soon as being approved by the end of this month. And you'll see a little more about that. But I think it takes a long time, and I think it's also risky because some of those technologies have not been tried before. And there are several already marketed antivirals, for as an antiviral, but they're being studied, and the remdesivir is actually authorized for use. If you listen to the news with President Trump, he was injected or given a drip for polyclonal antibody cocktail, which is being studied by a company called Regeneron. So quite a number of them are promising, but the disease has not stopped affecting common people. It's affected presidents of the world, the prime minister of the world, the home ministers of the world, the actors, the actresses, and everyone common to us. It hasn't spared anyone. It's amazing that this has happened. I think that's another reason why the speed of this discovery. So let's go to vaccine. And I'm looking at the time. So I'm not going to go to read all the stuff, given that we have only about 12 minutes more. There are two different vaccines which are going to be relevant for us. One is the mRNA vaccine. And these vaccines function by introducing messenger RNA sequence coded for a disease-specific antigen. Once produced within the body, the antigen is recognized and immune response is mounted. So if you look at, we'll talk about which vaccine is in the forerunner. So you have Pfizer, BioNTech, and Moderna, who have invested heavily on this vaccine. And you also find that many governments are actually looking at booked vaccines in before approval. And then you have the viral vector replicating, non-replicating vaccines, where Johnson & Johnson and AstraZeneca are involved in. Viral vector vaccines carry DNA into a host cell for production of antigenic proteins. They usually contain a live attenuated virus that is genetically engineered to carry DNA coding protein antigens from an unrelated organism. So different types, different risk factors, and you can go through the other vaccines, the DNA vaccines, the protein-based vaccines, and the inactivated attenuated vaccines. And if you really look at the bottom, I just picked a few companies you might be familiar with who are actually investing heavily on these types of vaccines. And we'll come to when will these vaccines be available in the market. So if you really look at this chart, it talks about phase 1 of trials, phase 2 and 3, phase 3 and phase 2 and phase 3, 1, 2 and phase 2 and phase 3. And each of this divide by the type of vaccine. For example, if you look at this quadrant, it's all mRNA vaccines. If you look at this quadrant, it's actually viral vector vaccines. And if you look at this part, pie, it's the activated, attenuated, this is a protein base, and these are the DNA vaccines. You'll see all these companies, for example, in phase 3, BioNTech Pfizer is in phase 3, and this is the company which came yesterday and said they're going to have the initial readout of phase 3 weeks from yesterday. You have Moderna, which is also in phase 3, and then you have J&J, and then you have AstraZeneca. So AstraZeneca is an important vaccine. So you must have heard that the phase 3 was halted because of a side effect, or what is known as an adverse event of one of the patients called transverse myelitis. And within a week, AstraZeneca was able to get approval from UK, South Africa, India to go ahead with the trials, except US. The US has asked AstraZeneca or AZ to come up with more data on why this side effect came up. And that's going to be important to watch. If you really looked at the timeline, AstraZeneca was way ahead of these people as being the number one, but it's not going to be the case. That's going to be an interesting thing to watch over the time. So who's going to be number one in terms of coming up with a vaccine? This is the data which was end of last month. And if you really look at who is going to launch, is BioNTech Pfizer. So this is the company which is going to be number one. and very quickly followed by AstraZeneca, and probably even earlier by Moderna. And the rest of the companies are J&J and Sanofi are very quickly behind. So according to this development timeline, the world is going to see a vaccine, an mRNA-based vaccine by next month. It's not that simple. You have, J&J with a viral vector, AZ with the viral vector, and then you see Moderna with a mRNA vaccine. There are some issues with that. I think Pfizer BioNTech uh, is manufactured in Brazil, in actually Belgium or Germany and the UK markets, in Ports, Belgium, and then in Kalamazoo in the US for the US market. And we'll talk about, and I think some of the characteristics of in terms of storage, we'll talk about it when we come. And efficacy is going to be early read in October. And the potential pricing, I think if you really look at what price this could be, this is relatively not so bad, given the fact that the governments are going to pay for this. And the response so far have been very good, except in J&J. We'll talk some of the characteristics. Look at the temperature it has to be stored. None of them are normal shelf temperatures. It's either 2 to 8 degrees. Pfizer, the BioNTech, which is going to come first, has to be stored between -70 and -90 degrees. And 2.8, 2 to 8, the AZ, and the -20 for Moderna. So these are the number of doses which are going to be available in 2021. There's going to be 3 billion patients in 2021 who, given the current production potential, can do. This still talks about only 50% of the world population. So hopefully by 50% or more being vaccinated, herd immunity is going to grow because it's 50% being vaccinated and people who already have antibodies. So that makes it, close to 60, 70%. Hopefully we will see herd immunity by middle of or early next year. And you can see the, from the Russian vaccine, even though Russia registered and approved without a phase 3, it's still, in US doesn't recognize the Russian vaccine because it has to go through phase 3 with a little more safety trials. And you have a lot more of them, and I think, hundreds of them in terms of vaccine. So the mRNA vaccine, which is going to be available in this month. So what is this all about? So mRNA stands, the messenger RNA stands for messenger ribonucleic acid, which is a molecule in cell that carries codes from DNA to make proteins. The mRNA vaccine encodes proteins of a virus, which is inserted into a cell to trigger an immune response. So mRNA vaccines are non-infectious because they don't have an active ingredient, active culture. The other thing is it's considered recursive because the technology is still new. What's great about this stuff is it can be produced in large quantities. Many other vaccines are, such as the flu vaccines, the egg-based, cell-based, or synthetic. So the interesting part, if in the US, people were asked in May of 2020, hey, if there was a vaccine available, would you take for sure? And only 72% said they would. Some said 42% said definitely, and 30% said probably. But when the AstraZeneca adverse event came in, that dropped from 72% to 50%. Can you imagine a pandemic like COVID-19 and a vaccine available? Only half the population is happy to take the vaccine. Even within the half, only less than 1/4 is willing to take the vaccine without any problems. So that talks about the synthetic nature or the manufacturing or the vaccines themselves. There's a big pushback on the vaccines part. People want to wait and see what happens if you take the vaccine. Is there going to be a serious side effect? So I think this is a good thing for the biopharma sector. There's a lot of trust that they would like anything coming out to go through vigorous testing. And FDA is known for it. And I think this talks about that anything new is going to be a cautious approach. But still 51% would actually take the vaccine. This is going to be interesting. I'll be interested to see if this survey was done in India, what this survey would look like. Again, another opportunity to look at if some of these vaccines make it to India, especially the AZ one, what will happen when some of them will take or will not take. But there are some key factors affecting the vaccine production. I think one of the things is safety and efficacy. I think people are going to see wait and see, and they're going to look for adverse events. Pharmacovigilance is going to be absolutely important. Reinfection and virus mutation. I think the problem here is that, the one which I'm working on. There is a initial dose given and that has to be followed within 21 days on day 21 for a booster dose. And we don't know how long the booster dose will last. So there's a lot of uncertainty of how long you're going to be immune from the virus. That will become clear. And who gets it first? If you look at only half the people, the production for 2021, only half the people in the world will get it. There's issues for counterfeiting. There's issues for a lot of working, pulling strings and things like that. I think the general consensus is the people, first responders are going to get first, like people in the hospital, nurses, doctors, healthcare workers, policemen, firemen, and probably the second list is going to be people who are immunocompromised, people who have cardiovascular events, transplants, people who are diabetic, who cannot fight this infection as good as the others are. So that's going to be that. There's going to be distribution challenges. I think here's the problem. Let's look at the BioNTech products, -70 to -90 degrees. Take India. Where is the facility where you can store that product between -70 and -90? None of the retail pharmacies can. Many of the hospitals cannot. It'll be probably many major teaching institutions. Look at the issue we had in demonetization and taking money from ATMs when that happened. There was long lines. Imagine the number of ATMs and imagine the number of hospitals with that type of facility you have. It's going to be even more appalling to figure out how this is going to be a distribution challenge for India. Manufacturing, the mRNA vaccines are easy, but how about the other vaccines? The one vaccine I'm working on is 10 doses per vial. So can you take 10 equal doses per vial? Do you have that many syringes to dispose of once that is done? Because you're looking at 1.3 billion vaccination the first time around in India, followed 21 days with a booster dose. Pricing pressure, with this economic burden, what will the pricing pressure look like in terms of pricing for countries? So I'm going to actually speed up my thing. I have only 5 more minutes. So clinical, if you look at drugs, there's 3,310 drugs on COVID-19 trials on trials today. It's being investigated, out of which about 1,386 are sponsored by a biopharma company. 605 from the US, about 328. I think this is a very, very promising slide, which tells there's a lot of work being done, but it's going to take some time in terms of figuring out the efficacy. And the type of drugs, we'll talk about it. These are the 5 drugs which are authorized for emergency use. Remdesivir is one. You can see that here, done by Gilead Sciences. And this is mostly this and the Regeneron drug, which President Trump has taken. It just helps you reduce viral load for people who can otherwise not fight it out. It's found to be efficacious at the early disease state, and this is authorized when the patient needs hospitalization. And you look at from phase 1 to phase 3 to phase 4, and you can see all the monoclonal antibodies and the antivirals. And I think there's a talk after me on the monoclonal antibodies. You can talk about what is their role in the COVID-19 treatment paradigm. And you talk about remdesivir as experimental medicine. Okay, this is the only thing which is authorized. None of them have never been authorized by the FDA. It's only authorized for emergency use in adults and children who are hospitalized. You need to be hospitalized for that and under the care of a physician. It's not approved, but it's in clinical stage. But in May, I think there was early results that the viral load comes down with this antivirals, and so it was authorized for that. And you can see a lot of combination drugs in terms of trials investigating. There's more than like close to 300 vaccines, probably way more, and a lot of trials. Hydroxychloroquine is the number one combo drug which is being studied. Followed by a lot of antivirals, azithromycin, and some of the antivirals. I'm sure a lot more monoclonal antibodies as well. This has had an impact on the other clinical trials. Unfortunately, because patients couldn't go, there was focus and there was issues on enrollment. There was a 67% decrease in patient enrollment in other clinical trials. If you look at the decrease in average patient enrollment, even respiratory disease, 34% decrease. Oncology, almost 50% decrease in terms of enrollment. This is putting a burden on the rest of the clinical trials. I'm not going to go through the devices. I think there is discussion tomorrow. One of the myth busters is the origin of COVID-19. Many people think it's in a lab from the US, a lab from China, but again, you cannot definitely say that because we don't have the data from China, China's lack of population. But I think this is also becoming political. But we all know it's from China, but we don't know exactly where the source is. We talked about the myth that it affects only old people. And India has proven it wrong. It affects only men, mostly men, and India has proven it wrong. So more studies are needed to find out, hey, is the impactable population a small population, or is it actually distributes about, based on region? The cure and treatment, many people think, hey, there's some homemade cures, and I've seen WhatsApp messages after WhatsApp messages how certain homemade remedies help you with fighting COVID. I think none of them are approved. It's all homemade remedies. And if you feel comfortable, I think it's your call. But I think very quickly this month, you'll see a vaccine come out, and very quickly thereafter, you'll see some of the drugs being approved. Needless to say, it's had an endless social economic impact. And the worst is yet to come. I think you'll see more and more what's happening, which is going to come. So what are you doing about this? I'm going to ask you the question before I end my talk. This is an ICMR study which came out, I think, a week ago, two weeks ago. More than 63 million Indians may have already contacted COVID-19, according to a new sero survey conducted by the ICMR, 10 times higher than official figures. Between mid-August and mid-September, the ICMR took blood samples from over 29,000 Indians in 700 districts across the vast country. 1 in 15 people over the age of 10 were found to have developed COVID-19. If this prevalence rate is applied to the 968 above age of 10 in India, it equates to 63.78 million citizens having been seen exposed to the virus. That 10 times more. Today, the official figures are close to 7 million. But according to the ICMR studies, it's 63 million. Is this true? Is there a differential on that? There's something, and it's worth in terms of epidemiology studies. There's so much to do with this virus. There's so much unknown. It's just a great place to start. The other thing is, I think for those of you who know me, I published an article in PharmaExec in the US. It's about COVID-19 and the evolving science of pharmacogenomics. Pharmacogenomics talks about how a drug is metabolized differently depending on the genome of the person. So I think another ripe area for research is looking at, how does the drugs which are in development COVID-19, can they be selective in certain types of population, women, younger, older, certain geographies? It's just a ripe area for another application of pharmacogenomics. So another thing is, I'm going to leave with this stuff. Everyone has a way to affect the population. This is a very famous poet called Maya Angelou, who once said, I have learned that people will forget what you said, people will forget what you did, but people will never forget how you made them feel. I think every one of us has a duty in this pandemic to play a role. And I hope you play a role where you make people, you don't say something or give something, you play a role where you make people feel differently. I'm going to stop. I don't know whether I have time for questions, but I think you can talk to your um, faculty in BITS Pilani, and they have my contact details. If you need any questions or any particular slide, please feel free to talk.
Thank you, Mr. Stevens. That has been an incredibly insightful discussion. We will be opening the floor for a couple of questions. If anyone in the audience could enter into the chat box.
That was a very fast presentation. I apologize for the timing, but I think there are way more questions than answers at this point, but I think feel free if you have anything in your mind to ask, and if I can, I will.
All right, we wait for a couple of more minutes. So there have been a couple of studies that have linked dengue and the contraction of the virus, and how people who have had dengue in the past are less susceptible to the virus. Is there any substance to these claims?
Yeah, I mean, there are several things which correlation doesn't lead to causation. So I would say, while that might be true, I think we need more studies, just not correlating, but actually finding the causation and the science behind it. The same thing with old people die faster because of immune response. That talks about correlation, but also a little bit about causation. So to the extent, my answer is, yeah, that could be true, but more studies are needed. We have to be very careful correlating and then saying that's the cause as well. So that would be my answer. But again, I'm sure the faculty or someone might have a different answer to that.
Right. There's a couple of questions in the chat box. Can the BCG vaccine help in preventing COVID, or can vitamin D help in preventing COVID?
Yeah, I mean, people have, again, same thing with correlation. I think that has shown, but I don't think that's true now. I mean, look at, if you look at ICMR, 68 million people affected, and I would find it hard to believe that 68 million people did not get the BCG vaccine. Again, that's something you need to study. More data is necessary, and I think that's where my first challenge is. I think the area is very ripe to answer some of these questions, and I think you have data coming in more and more. There's a small window you have before vaccines come in, and that's a very ripe window for you to figure out all these, test all these hypotheses. The question is, the other question will be like, how come some people are asymptomatic? If you look at the ICMR studies, that begs the question. There's quite a number of them are asymptomatic with the same range, same gender. Why is that different? So it's a virus. I think, to the fact that some of the existing antivirals have helped with the reduction of viral load. Some of your BCG vaccine, every vaccines, could play a role in terms of that could be happen. But I think, again, at this point, it's correlation, but not proven in terms of causation. So good question.
All right. Thank you, sir. Thank you to the audience members for sending in their questions. There are a few more questions, but I would encourage you to reach out to Mr. Stevens personally, as it's time to move on to our next speaker. Thank you, sir. I would also request all our participants to fill out the Google form that has been shared in the chat box. It is compulsory for all the participants. Our next speaker for the session is Dr. C.K. Katiyar. Dr. Katiyar is a renowned researcher, academician, and expert in regulatory policies for traditional medicines such as Ayurveda. After completing his master's in Ayurveda and doctorate in pharmacology, Dr. Chandrakant Katiyar has been at the helm of affairs at India's best research and development driven companies like Ranbaxy Laboratories, and Dabur India Limited in key leadership positions. Currently, Dr. Katyar is the CEO Technical of Imami Limited and is responsible for research and development and quality control of Ayurvedic and herbal drugs. With many years of experience in clinical trials on Ayurvedic drugs as well as synthetic molecules, Dr. Katyar is an expert on pharmacological studies and toxicological evaluation of herbal drugs. He has also worked on developing pharmacopoeial monographs of herbal raw materials for Indian pharmacopoeia and United States pharmacopoeia, and has contributed in intellectual property right protection of traditional medicines in India. Dr. Katyar has also initiated the development of phytopharmaceuticals, a new class of drugs in India, and is currently the vice chairman of the National Phytopharmaceutical Mission of the Department of Biotechnology, Ministry of Science and Technology, Government of India. He is a renowned author and has written several books, including Modern Ayurveda, Milestones Beyond the Classical Age. Sir, I request you to take the stage.
Thank you very much. Can slides, please. Is my slide visible?
Yes, sir.
Okay, thank you very much. So, good afternoon to all of you. And you are aware that these days Ayurveda has become a buzzword, and the credit goes to Government of India for promoting health promotive aspects of Ayurveda through through boosting the immunity through Ayurvedic products like Chavanprash, Shashwagandha, Giloy. Some of the products which were difficult to inquire are suddenly become very, very popular, and they are getting off the shelf very quickly. So I thought that today I'll be talking about Ayurvedic herb for COVID-19. But before talking about the herb, let me first tell you, a lot of you may not actually be aware that what Ayurveda actually is. So Ayurveda is a neither science of disease nor science of ease. It is a science of health, science of life, good life, bad life, whatever life. And it's had two objectives. One is the preventive healthcare, prevention, preservation of the health, promotion of the health, and if somebody falls ill, then to treat the disease. So prophylactic and then preventive, two aspects of life. Now, within 2-3 slides, I'll tell you the gist of Ayurveda, how nature and human body are same, and what is the harmony between them. So 5 basic elements, Prithvi, Dal, Tej, Vayu, Akash, they are responsible for 6 kind of tastes in the medicinal plants, and these 6 kind of tastes reflect themselves in 3 biological forces inside the human body, where Vata is responsible for all kinds of movement, Pitta is responsible for all kinds of metabolism, Kapha is responsible for all kinds of structural integrity of the body. So they are called 3 doshas. So 3 doshas are affected by the taste. Rest are the going deeper into it. So if somebody takes sweet food, sweet, it will increase the kapha but reduce vata and pitta. Like that, all the tastes have some or the other effect on the human body through these 3 people used to call humors. I would like to call them biological forces, vata, pitta, and kapha. As per Ayurveda, total population in the world can be divided in 10 types, and they are basically depend upon the vata, pitta, and kapha. They are psychosomatic constitution, and they are decided basis physical and mental traits. So if some of you are interested, just go to the Google and type out the Prakriti questionnaire. You find out your Prakriti. It will also tell you what's good for you in the health, what kind of disease you are prone to, what kind of food is good for you, what kind of food is bad for you. So it was sort of personalized medicine, personalized advices, personalized guidance are part of the Ayurveda. Something very interesting description. It was 1000 BC in Sushruta Samhita. It was very close description of Sushruta. If you look at the English one, it says sometimes a town or a city is depopulated by curse, anger, and all those things. And there is epidemic of cough, asthma, catarrh, fever, irrespective of the constitution, and peculiarity of deranged body humors happen. And people even get away from all kinds of seeds, carriages, riding animals, gems, precious stones. They keep away from all of them. This was described in 1000 BC in Sushruta Samhita. It was something very close to a description of COVID-19 we are saying today. And there was no treatment. So what they said, can we protect ourselves? They said, yes, prophylactic measures. What are they? They are say, do namaste. That's one. Keep away from everything. Do some puja part, means do some practices of penance. Clasp palms to the gods. Be spiritual, obedience, kindness, and follow all the rules of conduct which are good for the society. So these are, they also said that in these kind of pandemics, treatment does not work, only prevention works. And when Ayurveda described the healthy living guidelines, how to live 100 years, active life, they provided three things. Dinacharya, daily routines, Ritucharya, seasonal routines, and Rasayana. Now, Rasayana is a very important concept which is using today. The Ministry of Ayush is using this concept today. So, what is Rasayana? Rasayana is not a chemistry. Rasayana, Rasayana is a pleiopharmacological activities in one. So, the product enhances immunity, antioxidant, adaptogenic, memory enhancer, anti-aging, anti-anxiety, and anti-depression. All them in one is called Rasayana. So that's why Ministry of Ayush promotes immunity. So right now, today, what's happened is Ayurveda, immunity, and prophylaxis have merged into one. And probably, we don't believe it, but probably that might be one of the reasons of lowest mortality rate in India, because it's hardly to find out anybody who is not taking either Ayush Kadha, or he is not taking Giloy, or he is not taking Ashwagandha, or even homeopathic medicine for prevention. Now, when we say Rasayana, which is an immunomodulator, there are three kinds of things. One is Achar Rasayana, means behavior modulator. Second is dietary modulator, and third is herbal drug. So only drug will not work. It has to be supported with the diet also. It has supported with the behavior also. Like right now also, whatever, even if the drug comes, we cannot do away with the face mask. We cannot do away with other things. So therefore, all the things have to work to be used together to derive maximum protective benefits from Ayurveda. He said the single biggest threat to man's continued dominance on the planet is the virus. And this saying, old saying, has come true today. Now, if you look at the 7 steps of how this coronavirus is attacking the human body, these are the 7 steps. And with these 7 steps also, hidden are the 3 or 4 mechanisms of action for developing the drug out of them only. So either you can prevent the entry of the virus, or you can prevent the replication inside the body, or you can modify post-translational modification of proteins. These are the 7 different steps. Out of them only we can consider the mode of action. But this slide we will come to later. So if you look at the immune system today, so it's a counter attack by immune system, whatever with the virus or bacteria or whatever. So even today we know that it is our innate and acquired immunity which protects our body. So that's one of the reasons when Ayurveda says that use the rasayana drugs. Use the rasayana drugs. Rasayana means immunomodulator, immunoboost. I am using the word immunomodulator. Now let me share with something very important with you. One drug, if given in the higher dose, will actually suppress the immunity, and in lower dose will elevate the immunity. Even this is true even for ashwagandha, even for guruchi. People don't know it. So therefore, do not take too much. Take within that limited dose only. Right. So we will now discuss some of the Ayurvedic herbs for COVID-19, and I believe I am a strong supporter of literature of Ayurveda and have great trust in our ancient literature. So basis that, after going through literature, we identified that if a herb is rasayana in nature, it has Shwas herb property, and it has Jwar herb means fevers, prevent fever, and it is katu and thick thrust, which I told you, taste affects the body. So any product which is having these 5 things, then this product can be one candidate for research on the anti-COVID virus. And we have identified 8 herbs out of great literature survey. We could find 8 such herbs. Now we will discuss one by one. Ashwagandha, is a right now very selling very hot, and it has one important compound which of special interest to us, that's vitamin A these days. Looking at as I told you that Ayurvedic medicines, Ayurvedic pharmacology is different from current pharmacology, and Ayurvedic pharmacological effect derived from the taste of the product on the tongue, and that's how they affect the body. Siddhyalka, Rasagoda Virupak on the left side, and their types on the right side. So they have certain therapeutic benefits of their own, which are rasayana, balya, increasing strength, neurological disorders, antidepressants. But some of the products, these are the benefits which already have been scientifically evaluated. Huge publications are there on anti-stress immunomodulator activity. But the issue is, do they also have antiviral activity? Yes. If you look, vitamin A has demonstrated the potential as therapeutic agent to prevent COVID-19 due to reported interference in viral S protein to photoreceptor binding. So, vitamin A from ashwagandha is a compound which has shown some activity, and one clinical trial of it, I know at least, is currently going on ashwagandha on COVID-19 in India. If you look at the turmeric, now curcumin everybody knows, but despite having these compounds and despite having these Ayurvedic benefits, this product is very good for breathing problems also, allergy and breathing problems, besides increasing immunity. But when we look at the literature, we could not find very strong literature on the antiviral activity of this product, though our other activities have been scientifically evaluated, like immunity enhancer. When we look at amalki, that's amla, amlic acid, which is major component of chyawanprash, and chyawanprash is a very important product for boosting the immunity. It has a compound like gallic acid and ellagic acid. It's Ayurvedic pharmacology. As per Ayurveda, rasayana means rejuvenates the body and improve the appetite. It also has been studied on all other scientific attributes like immunomodulator, antioxidant, and gastroprotective. But there are no studies, at least I have seen, on the antiviral activity of this product. Now, this is the herb of today. It's also known as giloy. Giloy is very popular these days in India to protect from the COVID-19 virus. Its main component is stenosis peroxide. Its Ayurvedic attributes are these, and it is also known as to be rejuvenator, strength promoter, but it also is a very good product to bring down the fevers and enhance immunity. So, lot of studies have been conducted on its immunomodulator potential, And some of the studies have also been conducted on its activity on the influenza type viruses, on Tinospora. So Tinospora, therefore Tinospora has become very important product these days. Tinospora Nashwagandha for COVID-19 protection. Tulsi, Osmium sanctum, as everyone of us know, it is the first line of therapy against any kind of cough and cold in India. People start taking tea of ginger and tulsi. So a lot of studies have been conducted, not only immunomodulatory activity, also on the antiviral activity of Osmium sanctum. If you look at the second one, the Osmium sanctum extract can be included as preventive measure against COVID-2 due to its inhibition or replication of CO2 supported with the immunomodulatory feature. So it has immunomodulator as well as antiviral activity. But the issue is that all these activities have been conducted on a whole extract, not a single compound. Yestimadu Meleti licorice is one more product which has been highly recommended these days. And if you look at the glycyrrhizin, which is a compound of this glycyrrhizin, it has been studied in detail for its anti-COVID-19 activity. And actually in India, it was the first paper from India appeared in Current Science was on glycyrrhizin from licorice and glycyrrhiza. This is also part of some clinical trials recently by the Ministry of Ayush. This is a plant which is little known, very, very little known plant. Somebody was asking the question about the relations between dengue and COVID-19. So now this plant actually was developed as an anti-dengue product when I was embassy at the time. And after now Sun Pharma is conducting the clinical trial on the first phytopharmaceutical drug, which is under phase 2 for COVID-19 virus. And this product actually was for dengue virus, but the mechanism of action says that though entry mechanism are different, but once inside the body, the mechanism of replication is same. So the products which are working for dengue virus will also be working for COVID-19, and they have conducted the studies. And now the product probably is in late phase 2 in India, and it will be first ever phytopharmaceutical drug, which is a new category of drug in India in late phase 2. And this is based upon a substitute of this plant, not this plant exactly, substitute of this plant. Now, Andrographis paniculata is another product which is very well known for respiratory diseases and also to antipyretic, lower down the fevers. This also has been studies on the for anti-influenza activity on multiple in vitro models. And there are people in Gujarat who are taking this, what they do is they dip the leaves of this plant at night in a glass of water. In the morning, they take this water. And it protects them from any kind of cough and cold infection. A lot of studies have been conducted on Kalmeg and antipyretic activity also. This was a plant which about 30 years ago, I think not 30, more than this. It was, I think, 75 when the first paper appeared on Phyllanthus as a product for hepatitis B virus by Tyagarajan and Bloomberg. In Lancet at that time it appeared. It was working for the hepatitis B virus. And so a lot of work has been done on this product on hepatitis B virus, but there is some work which is still going on this same plant on the SARS-CoV-2 also. And initial results are encouraging, but let us see what happens when it enters into from preclinical to clinical stages. Mamira is a capristita. It's one of the fantastic source of berberine. Now, berberine has been, as a single compound, has been very active in bringing down the fever, in boosting the immunity, and in anti-influenza viruses. Currently, no product based upon berberine, based upon this Mamira is in the market. Neither this product is any stage of clinical trial. But to me, it appears that this can be one good candidate for clinical trials in COVID-19. As on today, there are some 43 clinical trials registered on clinical trial registry in India on Ayush products. Out of it, 31 products are registered at 88 centers, including one phytopharmaceutical I told you by Sun Pharma. That was a phytopharmaceutical drug. Now the question is, can there be a single herb-based product for SARS-CoV based upon research? As if we go by Ayurvedic principles, Ayurvedic understanding, no, this cannot be. Because the reason is that everybody had different psychosomatic constitution, one drug does not fit all. But the combination, may yes. So there are a combination of one or two herbs, probably may be there, which can work on everybody. Now, only drug also would not work. As we said that nowadays everybody says also that you have to support with the face mask and social distancing. So therefore, lifestyle measures, restrictions need to be followed with this also. And now coming up, remember, some of you might be remembering when you were kids at home, our mothers were not allowing us to take any food without washing the hands before having food and after having food, both ways, right? So I think all things are coming back again, and the new normal is bringing back some old traditional habits also, and it is also bringing back the faith and trust in the old traditions, including Ayurveda, which is the traditional Indian systems of medicine. And as on today, as I told you, 43 clinical trials are going on, and 80,000 Delhi policemen are currently being given one Ayurvedic kit to protect themselves from coronavirus, and 50 lakh means almost 5 million people have been given different kinds of Ayurvedic kit to protect themselves. And it is sort of observational study, and I think within 3 to 4 months' time, Ministry of Ayush will come out with the data of both. But I certainly feel that the lowest mortality rate of 1.6% only in India. Certainly, frequent and very popular use of Ayurvedic remedies like be it Ayush Kadha, be it Giloy, be it Chavanprak, certainly it is playing a role in not only lower mortality, but also in lower intensity of symptoms. So, therefore, the benefits of Ayurveda have, at least during this pandemic, Government of India has promoted the prophylactic part of Ayurveda, which was highly neglected so far. And that is the strength of it. So I am very happy that these Ayurvedic products are saving a lot of lives of the people, saving a lot of suffering of the people. And I hope that people should continue taking them along with the other restrictions like social distancing and face mask, etc., to derive maximum benefit out of them. With this, thank you very much. And if any questions, I leave to the organizers whether they want to ask right now or later. Thank you very much for the patient hearing.
Thank you, sir. There are a few questions in the chat box. So there's one from Dr. Suneet Shukla. In clinical studies with ashwagandha, what product is being used and the patient population that's being studied?
It's being used ashwagandha powder. Ashwagandha powder is being used, not the extract. And I think the patient population is being conducted at Banaras in the university. If I am not wrong, it's 100 or 150 patients only, because it's a CCRAS, Ministry of Ayush sponsored project.
Right, sir. The floor is open for a couple of more questions, so we'll wait for a few moments.
Sure.
I request the audience to fill in the Google form if they have not already. It is compulsory. Thank you. All right, there's another question. Our Ayurveda is having a huge potential in treating many diseases, but there is a lack of knowledge among young India. How do you identify spurious products and genuine products?
Certainly, I fully agree that it has a huge potential, and actually, its potential is much more in the disease preventive and health promotive area rather than, I will say, disease alleviation. And I think for the first time, it is being promoted for the right strength, which is the protection from disease and promotion of health. Yes, and as far as young India is concerned, young India is very, very adventurous in nature. They are now moving towards nature much more than the elder population like myself. So I think that young Indians will enjoy benefits much more. And now everything is available on the Google. So Google Baba is the best teacher, but sometimes it gives wrong knowledge also. So there is a need for more promotion of the product, but good thing that government itself is promoting the prophylactic effect or preventive effect of Ayurveda. So young India also has to move forward. They also need to do some literature search, do some interest on product like say when you are going karkuma, haldi latte, haldi milk. Now haldi milk has become very popular these days, right? Guglu Ayush Kadha. If you can replace your habit that you take at least once a day, replace 1 cup of green tea with 1 cup of Ayush Kadha, that itself will take care. So I think that applies.
There are quite a few more questions. If you could take a couple of them. Phytochemicals seem to have no side effects and are mostly non-toxic. So what is the hurdle in large amounts of trials for inhibition or prevention measures? A
No,
question.
phytochemicals are not allowed in use of single phytochemical is not permitted in Ayurveda. So there is a new class of drug called phytopharmaceuticals, which is fraction of an extract, and this fraction may contain hundreds or 200s of chemical compounds, but enriched with 3 or 4 compounds. So pure phytochemicals are not permitted either in Ayurveda or in phytopharmaceutical. If somebody has developed a drug out of pure phytopharmaceutical, you need to follow new chemical entity route. And that's a huge, altogether different subject.
There's another question from Dr. Sunit Shukla. For powdered in clinical studies, how do they ensure quality control properties of API?
Sure. So, like if you take a paracetamol tablet, you know it contains 500 milligram of paracetamol. It may be 490, it may be 510. But the question is right. If I am taking, say, ashwagandha 250 mg capsule, how do I know it has 250 mg capsule, right? So now science has advanced too much. Now we know that the ethanolite content in a compound from ashwagandha is say 0.2%, 0.3%. And if I take HPLC, I can quantify ethanolite content, I can calculate back. Yes, this capsule does certainly contain 250 mg. And these kind of quality control measures are nowadays mandatory. The pharmacopoeias are there. You'll be surprised to know even United States Pharmacopoeia today has 75 monographs on Indian Ayurvedic plants. I am a member of the committee. My lab itself, between Ranbaxy and here, have contributed some 30 medicinal plants to the United States Pharmacopoeia, where all these quantifiable parameters are there. You use those compounds, they are called marker compounds, and you can back calculate the actual quantity of earth. So quality control has now improved too much, and there are negative markers also, like for contaminants, heavy metal, pesticide residues, aflatoxin. All these negative markers, all these contaminants also are being tested now, part of the quality control manual by Government of India itself, even by Ministry of Ayush in India. So things have advanced too much. People don't know about.
All right. Thank you, Doctor. That's all the time we have for questions. Thank you for that insightful session. We'll be moving on to our next speaker.
Thank you.
Dr.
Thank you.
Thank you, sir. Our next speaker and our last speaker for the day is Dr. Suneet Shukla. Dr. Suneet Shukla is a pharmacist with a PhD in molecular pharmacology from the Jawaharlal Nehru University in New Delhi, India. He has completed his postdoctoral training at the National Cancer Institute, NIH, studying drug transporters and their role in cancer drug resistance, fungal infections, and altering ADME toxicity of drugs. He then joined the FDA and has worked at the Office of Pharmaceutical Quality and the Office of Generic Drugs, where his responsibilities included the assessment of INDs, NDAs, ANDAs, and other regulatory submissions from a pharmaceutics and bioequivalence perspective. Currently, he is working as a senior pharmacologist at the Office of Clinical Pharmacology, where he is involved in the assessment of IND and NDA applications in the area of immune and inflammation drugs from clinical pharmacology perspective. Dr. Shukla possesses years of highly specialized research and regulatory experience in PKPD data analysis, in vitro drug release and dissolution analysis, bioequivalence determination, IV-IVC model development and validation analysis for SUPAC changes or biowaiver applications, quantitative modeling and simulation analysis for small and large molecules from early to late drug developmental phase, drug-drug or drug excipient interactions using transporter-mediated IV-IVE PBPK model to understand drug absorption and oral bioavailability, DDIs, and pharmacogenomics in drug interactions. Dr. Shukla is on the editorial board of the Journal of Anti-Cancer Research and serves as a peer reviewer for several journals and has published several highly cited research and review articles and has received patents and scientific awards from the FDA and NIH. He has served on panels evaluating research grants submitted to NIH and FDA, has mentored several undergraduate, graduate, and postdoctoral trainees, and currently serves as the lead instructor of the medical pharmacology course at the Foundation for Advanced Education in the Sciences. I would like to hand over the mic to Dr. Shukla.
Hi, can you all hear me?
Yes, sir.
Okay, let me just try sharing my screen. Give me a second.
Sure.
Can you see my screen now? I don't think so.
Not yet.
Okay, hold on.
Yes, sir.
Can you see?
Yes, sir.
Okay. One more second. Is it on a slideshow now? I cannot see my own screen, but if you all can.
It's not on the slideshow mode yet, sir.
Oh, hold on a second. Let me just move one more time if it doesn't work. How about it? Can you see the slideshow mode now?
Yes, sir. Yes, sir, we can.
Okay, thank you. Thank you. Thanks to the organizers for inviting me to share my perspective. I'll not be talking about COVID and COVID-related drugs, but I will be giving you the basics of monoclonal antibodies drug development as it applies to both preclinical and clinical pharmacology, and also about the general aspects of drug development and about biosimilars. Uh, Let me just move. This is my disclaimer. What I am going to present today are my own personal views and should not be construed as the views of FDA or the US government because I'm a government employee. I have to put this disclaimer. So this is general outline of my talk. I'll introduce the basic concepts about biologics, followed by monoclonal antibodies and the basic concepts of preclinical and clinical development, which applies to development of monoclonal antibodies. And then certain concept of clinical pharmacology considerations, which should be put in there to develop these biologics. And the last part would be about biosimilars, how the biosimilarity works in the context of biologics. So biological products are regulated by the FDA, and as the case with the normal drugs, the biological products are also classified as the ones which are used to diagnose, prevent, treat, and cure diseases and medical conditions. They represent a diverse category of products and are generally large and complex molecules compared to small molecules. These products may be produced through biotechnology in a living system, such as microorganism, plant cell, or animal cells. And there are many types of biological products which are already approved in the US, including therapeutic proteins such as filgrastim, monoclonal antibodies such as adalimumab, and vaccines such as influenza and tetanus vaccines, and a lot more vaccines, including the COVID vaccine, which is considered a biological product. In terms of the products, as I mentioned, it includes vaccine, blood, and blood components also come under that categories. Certain allergenics, Botox, for example, somatic cells and gene therapy gene therapy products, and tissue and recombinant therapeutic proteins, they all come under the category of biologics in a broad category of biologics. When we talk about biologics, protein also come under that category and in a very broad concept which applies to FDA regulatory perspective. If there is a protein which has a molecular weight of greater than 40 amino acid, that will be considered as biological products. And also clinically, the chemically synthesized polypeptides are also, which is a amino acid polymer made of entirely chemical synthesis and less than 100 amino acid in size would be considered a biological product. US has a history of biological product approval for a long time. Recently, last, I would say 15, 20 years, it has come more into practice. But even if you go back to 1902, that's when the Biologic Control Act was enacted, where in response to, there were two incidents which happened, which led to 22 children dying for the tetanus vaccine, which was contaminated. And that's when the Biologic Control Act was enacted. In 1944, Public Health and Safety Act was enacted. In 1938, FD&C Act came in. So this is in early 90s. In the 1970s up to 87 and 2003, FDA divided the biologics and their regulation into different centers. Even today, we have two different centers, CBER and CDER. One controls the biologics, the other controls the drugs. But under CDER, the monoclonal antibodies and therapeutic proteins comes under CDER. In 1980, as the FDA approved first genetic engineered insulin, which is a biological product. And as of March this year, insulin has come into this biological product pathway. So now it is not considered an NDA as a new drug application. It's considered as a biological product. So biosimilar products can come into market. Then in 1986, the first monoclonal antibody, Muromonab, which is a mouse antibody, OKT3, was approved. Coming back to last 10, 15 years, there has been a significant progress in terms of the biologics approval in US. In 2012, the Biological Product User Fee Act was enacted, where now FDA has this user fee associated with the biological approval that gives industry an advantage of having a fast-paced approval by paying money to the FDA. From 2015, there was a revised guidance. There was a draft guidance. 2019, we put a CMC guidance for biological products. 2017, there was a Clin Pharm guidance for biologics. 2019, we had an immunogenicity guidance. And 2019, we also had an interchangeability guidance. So all in all this work was done under the BPCI Act, which is meant for introducing biological products into the US market. And then since 2010 until 2015, there are many antibodies which have been developed and approved through this biological product pathway. And we have many more biosimilars already in the pipeline, which are being approved. So we do have lots of biologic regulatory history and activity going on currently at the FDA. In general, how the biologics are different from the chemicals, the small molecules. It's a very simple schematic here where chemical medicines as such, those small molecules, they are chemically synthesized drugs. You basically have to add some chemicals, and then what you get is an API which has a very defined structure, and then it can be characterized from quality control perspective. On the other hand, biologics, they are grown from living things. They must be manufactured under precise conditions, following exact steps, and they should yield a consistent product. And they are highly sensitive to manufacturing conditions. For example, if you clone a DNA and just put it into cells to produce protein, depending on the cell type, the protein which is being produced may be different. And because of that, the end product, which is a biologic product, would be a different product. Because of that, small molecules versus large molecules, as I call they may have different properties. So small molecules are very easy to control in terms of their characterization at the molecular level. For biologics, you need much more evidence, and that's a very big difference. That's why they follow two different regulatory approval pathways for the drugs. This is another schematic, just to put this in perspective. This is the size of a monoclonal antibody versus size of a small molecule, which is aspirin. As you can see in the picture and shown below, a single monoclonal antibody weighs more than 800 times what an aspirin molecule weighs in here. So you can imagine the complexity which goes behind the characterization of aspirin versus a monoclonal antibody. And it's a lot more work in terms of making sure that we have the right product which goes into the patients. This is another comparison, basically telling you what I just said earlier. Small molecules are generally low molecular weights. Biologics are high molecular weight. They are made by organic or chemical synthesis versus the biologics are made with live cells, and they have inherent risk of contamination. Small molecules have critical processes fewer critical process steps, while biologic products have many more because of the processing, both upstream and downstream. Small molecules are well characterized, biologics are less easily characterized. Small molecules structurally, you can identify structure relatively easily, but for biologics, that's very difficult. Also, they are homogeneous drug substance for small molecules. Biologics, as I said, they may have heterogeneity associated with it because of the way they are grown in the cells, and that includes variants, and that's a big issue from quality perspective. Small molecules generally are not immunogenic, but biologics, they are also immunogenic, and that's another area of research which is very active. Anytime a new biologics come into it, the immunogenicity is a big concern, and we are always worried about it. So that was introduction about biologics per se. Coming to monoclonal antibodies, and this is one category of biologics which are actively being used, and I would say more than 70% of biologics which are approved currently, are monoclonal antibodies. This is a general structure of a monoclonal antibody, and it has certain elements to it. Antigen is the one which is the toxin or other foreign substance which induces an immune response in the body, especially the production of antibodies. And the antibody, as shown here, is a blood protein which is produced in response to counteracting a specific antigen. Now, this antibody has specific elements to it which are responsible for the activity of the antibody. FAB region is the fragment antigen binding region of the antibody, which is here. That is responsible for binding to the epitope on the antigen, and the corresponding region on the antibody is called paratope. FC region, which is at the bottom, is the fragment which is also called crystallizable region. It's in the tail region of an antibody. It interacts with the cell surface receptors called FC receptors and some proteins of the complement system. Now, these antibodies have two different chains, heavy chain and a light chain. And then depending on the type of the heavy chain, which I will describe in my next slide, you classify the antibody. The hinge and domain, which is interdomain region, is also important for the activity, and also certain products which are manufactured by taking the hinge domain, by cleaving the hinge domain, so you only have the FAB region. And then idiotype is the one which is a unique set of antigenic determinants outside of the antigen binding site. So in general, human immunoglobulins classes are basically identified by the heavy chains, and these heavy chains are designated as Greek letters, alpha, gamma, delta, epsilon, and micron, and the immunoglobulins are produced, therefore, are called IgA for alpha, IgG for gamma, IgGD for delta, IgG for epsilon, and IgGM for micron. And each immunoglobulin isotype carry one type of a light chain, as I showed in the previous slide. It could be the kappa or lambda light chains. So, for example, if we say belimumab is an IgG1 lambda monoclonal antibody, so it's an IgG type of antibody, and the lambda is the light chain here. On the other hand, belimumab is IgG1 kappa, where the light chain is the kappa antibody, a kappa light chain. This slide, I don't think we need to go again, but the point I want to emphasize here, because it's a complex molecule, it has these variable region, constant region, and then there are effector functions, which are mediated by either the FcRn part or Fc gamma receptor interaction. It is much more work in characterizing the antibody activity, especially from the clinical perspective as well as the quality control perspective. This is another slide of what I said earlier. The FAB region in the antibody is responsible for binding to the antigen, and this binding is very specific, and that's how you design a specific interaction for antibody for its antigen. On the other hand, the Fc region of the antibody, it binds to the Fc receptor on the cells, and it's a non-specific type of binding. These two types of bindings are mediated by cell surface receptors or receptor target, which are in solution and are both in combination. And the good thing about the antibody binding with the antigen, which is mediated by FAB, is the affinity, which is very high affinity. It goes into picomolar affinity, and that's where the specificity comes into picture. For the sake of time, I'll skip this slide. I will go into the preclinical and clinical development of the antibodies. As it goes with the small molecules, the similar principle applies for the development of large molecules, except that the type of studies which are done are different, and the quality attributes also are different for small molecules. So as you can see, the discovery starts, and then most of the CMC step of work in terms of the quality attributes which are related for the antibody activity with the target are done at this stage, and then follows by the non-clinical studies where in vitro and in vivo pharmacology of those biologics for the antibody would be done on each target. And that also happens in discovery, but discovery and non-clinical, these two things sort of goes hand in hand in terms of looking at the quality attributes and also making sure that there is no non-clinical effect which is inhibiting taking the progression of the antibody to the next stage. The tox assessment is done during the non-clinical part, and the relative potency is compared to the monospecific comparator, which is the current standard. It will mostly be done in the non-clinical part. Then comes the phase 1 studies. In the phase 1 study, the first study in general, as it applies for the small molecules, is the first in human studies. And I'll discuss this a little in little detail a few slides later. But phase 1 starts with first in human, which normally is a single sending dose or a multiple sending dose followed by a single sending. The relative bioavailability, special population is done at this stage. Phase 2 generally is done in the target patient population, where it's a proof of concept study, and you mainly do the dose ranging studies to identify a dose which will be used in the pivotal safety and efficacy trial, which is phase 3 trial. And based on the data from phase 3 trials gets approved, then it goes into the life cycle, where you either have the post-marketing commitment or requirement depending on the biologics, and the next component is the pharmacovigilance after the drug is approved. This is basically giving you the same path, but looking at the stages, these are the objectives and the studies which are done, as I said on the previous slide. From the clinical perspective and from pharmacology perspective, what is important is identify early PK liabilities using the preclinical models. That's something which is important for us as a clinical pharmacologist to know. That's something which is not going to interfere during the clinical development. And then going to the dose ranging PK in the species which are relevant, and then the pilot PK and PD safety studies, which is done in the appropriate species, mostly non-human primates or rodents. And that is also an important concern if your biologic is supposed to be active for a specific target. In human, that target should also exist in that non-human preclinical species as well. Certain part of the PK/PD modeling is done during this stage, but from a pharmacology perspective, these things are important, which leads to the IND enabling studies. And then after that, the pharmacometrics approach also are used to identify covariates, and some of this I'll cover a few slides later. I'll skip this again. This is basically similar strategy, but the point with the BLA filing, which is the end of the phase 3, is it's very important to have all these things characterized. And a lot of this is applicable for small molecules as well. But for biologics, it is a big issue. Coming to the clinical pharmacology considerations, which are important for the development of biologics, is the first time human dose selection. This is done to, first of all, you should have an identified pharmacological effect in animal models, and also a dose which shows no observable adverse effect levels, which we call NOEL. And these drugs, this dose is the dose at which you don't have any adverse event. For biologics, for certain class of biologics, it may not be possible due to very high specificity, and because of that, is very few off-target binding effects to have Noel level observed. But in terms of once you have the Noel, then you have to establish an adequate safety margin for clinical administration, and this should be clinical the preclinical studies, as I said earlier, it should be conducted in a clinical relevant species. Safety margins are calculated using multiples of the human dose, which is related to the PK exposure data. And then the safety margins vary with product class and the clinical indication. For example, for oncology, we may be okay with a lower safety margin, but for example, RA, rheumatoid arthritis, that's not acceptable. And once you have the human equivalent dose, then that would be the safe starting dose, and that would be something which would be escalation for the next clinical trial. Sorry, that would be the first human dose. Then that convert the dose which you have, the Noel dose, it gets converted into human equivalent dose that applied safety margin, and that is used for the actual first in human dose. Sometimes we also use minimal anticipated biological effect level. As I said earlier, if you don't get NOEL in animal model, you have to use an alternate model, which is this Mabel dose. That is the dose which is mainly used based on the pharmacological effect, and that is a minimal dose. Generally, the dose between Noel and Mabel, whichever is lower dose, that's recommended for first in human trials to start with for biologics. For dose selection trials for phase 2 and 3 studies, there are many factors, and I just try to put a couple of things in there which are important from clinical pharmacology perspective. Depending on the dose ranging studies which have been done after in phase 2, phase 1, or that's the one which would identify the dose which will be used in phase 3. Of course, there are these two different types of observations. A dose could be linear on the nonlinear PK. So mostly we try to stick to the linear PK part of the dose, which would be good for the actual phase 2 or phase 3. Then also about target occupancy, we look at the clinical at the dose which is used in phase 2 or phase 3, what is the target occupancy in there, and also exposure response relationship associated with dose. And based on the target occupancy and exposure response relationship, and the dosing is determined for the dose selection for phase 2 and 3. QT assessment, which is basically the QT prolongation, is very important for small molecules because they generally can enter the cells and block these human channels, which are responsible for having this QT prolongation event. And that was one of the concerns with hydroxychloroquine, which was approved for COVID. Yes.
So sorry for the interruption, but we actually have a few participants who are trying to draw on the screen. I request the participants to not do so, as it is distracting for all other attendees. Any participant who continues to do so will be removed from the meeting. Thank you. Sorry, sir. Sorry for the interruption.
No, thank you. Yeah, thanks for doing this because it is a distraction. Yes. Okay, so QT assessment is not a concern. It's not specifically mentioned in the ICHE 14 guidance, which we follow for biologics, but it is something which is a concern for small molecules all the time. As I said, there is very low potential to block the human ERK channels for monoclonal antibodies. That's why, because these antibodies are large molecules, usually remain extracellular. They cannot access the channels from the intercellular side, which is the target site for most small molecules. That's why you get this toxicity, and hydroxychloroquine is an example for that. TQT study interval is generally not required, unless there is a specific signal in the study. A routine ECG is monitored in phase 1, 2, and 3 clinical trials with biologics, but we don't go to dedicated QT study. In terms of the drug-drug interaction, which is also important from both small and large molecule perspective, for biologics, we do worry about the DDI as it relates to their effect on small molecules. As you can imagine, biologics are given mostly to subjects who have complicated health situation where small molecules were not able to provide them the relief. That's why mostly these patients are not on single biologics. They are taking small molecule drugs at the same time. So from clin pharm perspective, this is a big concern because these biologics can have effect on small molecules as it relates to modulation of drug metabolizing enzymes. For example, interferons, cytokines, they decrease the CYP mRNA levels and enzyme activity. That's why the small molecule levels may change. And also the biologics can have effect on transporters such as P-glycoprotein, and that can result in the increased bioavailability of certain drugs, in this case, digoxin, which could be a bigger problem compared to the actual effect of the biologics. Also, the effect of small molecules on biologics is also something which is typically we study in the context of immunogenicity. They can decrease the immunogenicity, such as certain patients who are taking, for example, methotrexate as background therapy with biologics for RA, rheumatoid arthritis treatment. They will have a decreased immunogenicity because of the small molecules. So those type of events have to be considered in the drug development for biologics, especially monoclonal antibodies from drug-drug interaction perspective. Another concern from clinical pharmacology is the special patient population, patients who may have hepatic or renal impairment. For small molecules, that's a big concern. But because of the molecular size constraints for the biologics, which generally are above 70 kD molecular weight, as I said in my introductory slides, these biologics, they have a minimal concern because they are not eliminated through the renal excretory pathway. That's why the renal impaired patients are generally not required for such studies. Also, biologics are not mainly eliminated by liver, so therefore hepatic impairment is not likely to affect exposure of biologics. Dedicated PK study for hepatic impaired patient is generally not required, but the applicants or the sponsors are encouraged to use the population PK approach to know the effect of biologics in hepatic impairment. Coming to the last topic, which is biosimilarity, and this is a buzzword now, and it's something which is FDA especially is investing heavily in terms of approval of biosimilars, which is basically the generic biologics, as you can put in other words. For people who are aware about generics are the low-cost alternatives to the small molecule drugs, and they have a pathway where you don't have to go through all the clinical studies. You just have to do a bioequivalence study, and then you can demonstrate bioequivalence and your generic gets approved. The similar concept applies for biologics and for biosimilars. And this was enacted in 2009 through the Biologics Prize Competition and Innovation Act, BPCI Act. It was passed in 2010 under Obama administration as part of Affordable Care Act. And this act creates an abbreviated licensure pathway similar to the generic pathway, but it is specifically for biological products, which are shown to be biosimilar or interchangeable with an FDA licensed reference product. So it's the same path, except that for generics, it's the reference product is an NDA. For biosimilars, the reference product is a BLA product. In terms of the concept, biosimilar products or biosimilarity, they are highly similar to the reference product, notwithstanding minor differences in clinical inactive components, which are basically formulation differences. And also there should not be clinically meaningful differences. And I would emphasize this, there should not be any clinically meaningful differences between the biological product and the reference product in terms of the safety, purity, and potency of the products. So this is the first step where you show the biosimilarity with a reference product. Then the next step comes the interchangeability. So once a biosimilar is approved, it can also be an interchangeable product. So the first criteria for being an interchangeable product, it should be biosimilar to the reference product as I said above. And it can be expected to produce the same clinical result as a reference product in any given population. But the last point here, that for a biological product that is administered more than once to an individual, the risk in terms of safety or diminished efficacy of alternating or switching between the use of biological product and the reference product is not greater than the risk of using the reference product without such alternation or switch. This is a complicated language, but basically it says that if you are doing an interchange between a biosimilar and you are doing a repeated change, the interchangeable product should not have any more or less risk than the biosimilar product. In terms of a basic concept, a biosimilar product can be prescribed to you by a physician, but an interchangeable product can be interchanged at the pharmacy level. A pharmacist can change that. So we still are not to a point where we have an interchangeable biosimilar yet, but we do have a lot of biosimilars approved. So the importance of the biosimilarity and interchangeability product is important. So I think I explained this earlier. It's an abbreviated license pathway for biological products. It has to be a biological product has to be highly similar to an FDA licensed biological product, which is publicly available information on the FDA previous determination that the reference product is safe, pure, and potent. Also, this licensing pathway, the biosimilar pathway, it's licensed under 351K of PHS Act. Here, you don't need a full complement of product-specific preclinical and clinical data because it's an abbreviated licensure pathway. You basically need a set of data to show the similarity to the reference product. A general requirement is a biosimilar product should be similar to the reference product. It should have the same mechanism of action. It should have the same conditions of use as has been previously approved. Should have the same route of administration, dosage form, and strength, and it should meet standard in terms of safety, purity, and potency. So it's the generic version of the biological product. For the sake of time, I'll skip this slide here. The goals of the standalone product and the biosimilar products are different. So standalone product, which is developed under the 351A path, which is the biologics path, the goal of that product is to demonstrate that the product is safe and efficacious. And the drug development generally starts with the preclinical development, as I discussed earlier, and it moves to phase 1, 2, and 3 pivotal trials to show the safety and efficacy. The important part is that in a standalone biologics, you have to demonstrate safety and efficacy. On the other hand, for the biosimilar, you don't have to demonstrate safety and efficacy. You just have to demonstrate biosimilarity between the proposed product and the reference product. And the goal is not to establish the safety and effectiveness of the proposed product. The goal is to demonstrate the biosimilarity. So FDA has outlined a stepwise approach where basically you identify the residual uncertainty as each step of making sure that your product is, the biosimilar product is very similar to the approved product, adapted product. And it's the totality of the evidence which is used for determining the biosimilarity. There is no one pivotal study that demonstrates a biosimilarity. So this applies a stepwise approach. And basically, the first point is the residual uncertainty, which makes sure that if you are applying, and it starts with the analytical similarity, then you go to a point that you look at what differences have been observed and how best to evaluate the differences, the potential impact of those differences, and what studies will be needed to basically reduce the residual uncertainty, basically making sure that the two products are very, very similar in there. So there are comparative PK, clinical PK and PD data, which is required to demonstrate biosimilarity. And the underlying principle is that they should have a similar exposure in terms of the PK and response in terms of PD. And if they are the same, then it provides similar efficacy and safety. So basically, you should have a similar exposure response relationship for the approval of the biosimilar. Clinical PK data generally will be expected. PD data is desirable, but it depends on the type of the biosimilar. As I said earlier, immunogenicity is a big concern, and it basically starts with phase 1 study. You have to monitor the immunogenicity of the biosimilar, basically in all phases of the clinical trial. Comparative clinical study is after the PK, if there is a residual uncertainty of the PK similarity, then you do comparative clinical studies. It's to demonstrate that there are no clinically meaningful differences between there. And it's based on the concept of bioequivalence, not bioequivalence, equivalence basically. So your product should not be superior or inferior. So basically non-superior and non-inferior to the reference product. Study population can be same as what is used in the actual clinical trials, or it can also be different. And then PK assessment is important for demonstrating the PK similarity, biosimilarity. So in summary, in terms of the biosimilarity, demonstrating biosimilarity is different from a standalone product development. The approach and the development program should and will be different based on the intended outcome to demonstrate biosimilarity. Analytical similarity data is the foundation of biosimilar development. And I think I've missed one slide here. Apologies for that. That shows the residual uncertainty demonstration starts with the analytical similarity data. It is very important for demonstrating biosimilarity. So understanding this relationship between quality attributes and the clinical safety and efficacy profile, it aids to the ability of determining the residual uncertainty about biosimilars and the predicted clinical similarity from the quality data. The nature and scope of clinical studies depends on the extent of residual uncertainty about the biosimilarity of two products after conducting an extensive analytical similarity assessment. Comparative clinical studies are necessary to support a demonstration of biosimilarity if there are residual uncertainties. A scientific justification must be provided to support extrapolation for the conditions of use. We have not discussed this, but that's also an area which is actively being researched these days in terms of extrapolation of biosimilars. And the content of biosimilar development program is based on the stepwise development and approvability, and it's based on the totality of the evidence submitted by the sponsor. I think this is my last slide here. I'd like to acknowledge my division members and my leadership here. And thank you so much for your time. And I can take a couple of questions depending on the time. I think we are still on time. So yes.
Thank you, Dr. Shukla. That was very insightful. There is a question in the chat box. I encourage the audience to send in any more questions that they may have. From Dr. L. Ramya. In case of autoantibodies, when there are no conformational or mutational changes in the epitope region of autoantigens, what can be the other factors for the production of antibodies?
Let me read the question again. One, in case of autoantibodies, is it in the context of the immunogenicity? I believe, yeah, it is. I believe it is the context of immunogenicity. It's not clear to me what the question is here. I can tell you a very general response for immunogenicity of antibodies, especially for biologics. You start with looking at the type of immunogenic response which you get with at a preclinical stage, even at phase 1 stage, and then depending on the response level, you go ahead to phase 2 dose ranging studies and try to maintain a balance between the immunogenicity aspect versus the dose which is being given.
Dr. Amir has said that yes, mainly in neurodegenerative diseases.
Yeah, so yeah, I think that's all I can say. In terms of the immunogenicity, we are being very careful starting with the phase 1 studies and looking at the response. The dose will be optimized based on the type of immunogenic response which we get.
All right, thank you, sir. If there's any more questions, do send them in through the chat box. We'll wait for a couple of more moments.
Okay.
Another reminder to fill in the Google form for attendance. As there are no more questions, I think this brings us to the end of our session. Thank you, sir, for your time. I would also like to thank our other speakers for today for their time.
Is Dr. Stevens online? I have a question for him.
I don't see him in the participants list, unfortunately.
Okay, that's fine. Thank you.
Thank you, sir. Thank you for your time.
Thank you all for inviting me.
Thank you, sir. With that, we come to an end of day 1 of the webinar. Again, thank you to all our participants as well as all our speakers for taking time out. I would like to remind our participants about the IDEA Challenge, which is sponsored by Visit. This provides our participants a platform to take their learnings from the webinar to the real world. They are invited to identify a single problem statement from those discussed throughout the webinar and propose a business plan solution. It is compulsory for participants to attend all sessions of the webinar to stand a chance to participate in the competition. I would, as a closing note, like to remind you that we will commence the 3rd session of the webinar at 11 a.m. tomorrow. This session will be on medical devices and the software sector and will feature distinguished speakers like Mr. Vibhav Garg, Mr. Vijay Simha, and Mr. K.S. Babu. With this, we will be wrapping up today's session. Thank you for attending, and we hope to see you tomorrow.